Okay, we know that the sequence of bases in DNA is converted into a sequence of amino acids in a protein by transcription and translation. But not all DNA codes for protein. Now, if you think about prokaryotes, so for example, let's have E. coli, bacteria, gut bacteria, but it's also been used extensively in molecular biology experiments. It's very well characterized. And about 80% of the DNA in E. coli does code for proteins. Okay, but if you look at eukaryotes, let's look at a human being. Right, we have 3 times 10 to the 9 base pairs of DNA. Let's say and then the current estimate is we have about 20,000 genes. And take into account the average size of a gene, that is less than or about 1% of our DNA actually codes for proteins. So the question is, what is the rest of it doing? Well, a small amount of it is going to code for RNAs, yeah? So if you think about ribosomal RNA and transfer RNA, they've got specific sequences they need to be coded for, but that still doesn't account for very much at all. Right, now, when we look at this value here of 20,000 genes, well, the human genome is, of course, entirely sequenced now. And what people have done is they have used computers to search the sequence for so-called open reading frames. Now, an open reading frame is something that codes for a protein. So it's got to have a start codon, which of course is AUG. And then it's going to have a sequence of bases, and then it's going to have a stop codon. And that's how this value of 20,000 genes has come about, by looking for these open reading frames. But let's just have a look at the structure of a gene, because even within an open reading frame, not all of it is coding. So let's have a look at the gene. So this is a so-called flanking sequence. That's not part of the gene. And then we've got, right, the start codon will start there, yeah. These red bits actually code for proteins, and these are called exons. But in between those exons, there are regions in blue there, which are called introns, and those sequences don't make the way into the final into the final messenger RNA in the protein. They don't. So there's the DNA. So what's going to be made of that is going to be, you've got initial mRNA transcript, which I'll draw. So I've got 4 exons and 3 introns in that gene there. So there's the mRNA. So that's the initial transcript, but that's not what is translated. What happens is that the introns are spliced out. So you end up with, right? And there's 3 introns which have been spliced out there. And that's the mature mRNA, and it doesn't make its, they don't make their way into the protein. Well, what's the purpose of introns? Well, no one's really sure. Some introns do control the rate at which the gene is transcribed. So, of course, all proteins are not transcribed all the time. So the most really obvious example, insulin. Insulin should only be transcribed in the beta cells of the pancreas. So it's under the control of every gene is under, the expression of every gene is under a lot of control. Sometimes they're switched on, sometimes they're switched off. And there have been suggestions that these introns have been involved in that. And another thing about introns is that it gives the possibility of so-called differential splicing. So if we look at this gene here, this mRNA here, we've got 1 2, 3, 4 exons, and quite often a cell can make two versions of the same protein. So it could make one version which has got exons 1, 2, and 3, and exon 4 there, that has been spliced out and thrown away. Or at other times it may make a slightly different protein where you've got exon 1, 2, and not 3 this time, but 4. So you can get two different protein products from the same gene. It's kind of a modular approach to making proteins. Different bits can be bolted on or taken off. So that's some of the perhaps possible reasons why cells have these intron sequences which are not transcribed. So that accounts for some relatively small amount of DNA which is not which is non-coding. Right. What else is it doing there? So there's an awful lot of DNA, and it doesn't appear to be doing anything. It's not an intron. It's not. It is not RNA. It's not tRNA. So what is it doing? Well, let's just talk about the structure of chromatin. So chromatin in eukaryotic cells, in prokaryotic cells, DNA is just naked, but in eukaryotic cells, it's not. It's mixed up with protein. So here, I've got they're supposed to be proteins, and those proteins are called histones. And each one of those balls is called a nucleosome. There's actually 8 histones in there, and the DNA is kind of wrapped around that. Like that, okay, and the black is the DNA. That's the structure of chromatin. Now you can the histones and the DNA, they can be combined. Either it can be quite a tight association, or it can be looser. Now, if it's tight, it means that you don't get much transcription. So the gene is switched off. If it's loose, it gets transcribed, it's switched on. And so some of this non-coding DNA, obviously all associated with histones, and whether that is tight or loosely bound to the histones is going to affect the degree to which the genes are expressed. And that, you come across this later, topic of epigenetics. And how the DNA is packaged it can alter the expression of the genes. And of course, in different parts of a cell's life cycle, it will have some genes switched off and some switched on. So if you think in an embryo, those cells can develop into any other cell, whereas in an adult, a skin cell can only turn into another skin cell. So the packaging of the DNA there affects its transcription, affects cell differentiation. Okay. Now, what we needed to talk about before we finish is there's a very large proportion of the DNA, maybe as high as 80%. And a lot of it seems to be it's fairly repetitive, and it doesn't code for anything. No one is sure if it does anything. In the 70s, it obtained the name junk DNA. And some people suggested that it doesn't do anything. It's just there for the ride, and it gets copied, and it's like a parasite, if you like. It doesn't do anything. That idea has fell out of favor. People tend to think it does do something. We're just not entirely sure what it does. But let's just talk about some of these highly repetitive sequences. You'll find highly repetitive sequences at the telomeres. Now, the telomeres are the end of the chromosomes. And they're needed for how the DNA is replicated. It's essential for that. You need these repetitive sequences there. There are other bits scattered throughout the DNA, these so-called transposon elements. Okay, and they are repetitive and they're found all over the DNA, and no one knows entirely what they do. One of them is called the Alu Transposon. It's 300 base pairs long. No one is entirely sure what it does at all, but there are about 10 to the 6 copies of that in the human genome. And that means it takes up about 17% of our genome. It's just this one sequence that really doesn't seem to do anything. So it is quite mysterious what a lot of this non-coding DNA does. Just to finish off, let's just think about there's quite a good argument that the non-coding stuff does have a function. If you think about a mouse and human being, we've obviously got the same common ancestor. We diverged about 75 million years ago. So that's a lot of time for evolution to occur in that period. But if you look at the non-coding regions, we are about 80% the same. And the argument is, if it didn't do anything, if the non-coding region was totally unimportant, 75 million years of random mutations and so forth would have made that an awful lot different than the 80% that it is. The fact that it has stayed the same is maybe a suggestion that yes, it does have a function, and any mutations to that cause it to change, have a deleterious effect on the organism, so it must be doing something.